This protocol is an easy and inexpensive alternative to using Ethidium bromide for DNA detection during electrophoresis. By removing Ethidium bromide, either UV light or blue light can be used for detection. Blue light is not damaging to DNA, which improves the chance of success for downstream experiments.
It's really easy to try this out. You just have to replace the Ethidium bromide in your gels with Thiazole orange.
To begin, prepare 1% mini gels by mixing approximately 0.7 grams of agarose in 70 milliliters of tris acetate EDTA or tris borate buffers. Next, dissolve thiazole orange in DMSO to make a 10,000x stock solution. While not particularly light sensitive, store thiazole orange in the dark when not in use. For long-term storage, make aliquots of the thiazole orange DMSO mixture and freeze them. Then add thiazole orange to a final concentration of 1.3 micrograms per milliliter, and microwave the mixture of agarose, buffer, and thiazole orange to dissolve agarose for approximately 60 seconds. Pour the agarose mixture into a gel casting apparatus containing an appropriate comb, and allow the agarose solution to solidify into a gel. To load the gel, first place the gel in the electrophoresis apparatus, if not already present. Then add TAE or TBE running buffer to cover the surface of the gel. Next, using a loading die, load 10 microliters of a DNA sample. Include a DNA sizing ladder for reference. Attach the cover and electrodes, and run the mini gel at 100 volts.